webinar. For the last 5 months or so, we have been living in an unfavorable and unavoidable situation. Surviving every day with fear is associated with some new words: pandemic, lockdown, COVID-19, sensitization, mask, infection graph, swab test, social distance, etc. We have felt the need for experts to shed light on various aspects of this topic, as we are constantly being enriched and burdened by the shock of new news, information, and opinions. Today's event is based on that thought. We are hopeful that this two-day discussion cycle will scientifically define and improve our myths, ideas, and fears also. In the near future, we will be able to live a much better life with the outbreak of corona or other infectious diseases. What we are getting to get extra in today's session is to know more about flow cytometry instrument and its widespread use on courtesy of Sci-Tech Biosciences, USA. Cordial respect and heartiest congratulations to all once again. I have the privilege to transfer the baton of this webinar to our Honorable Patron and Principal, Dr. Tapan Kumar Dotto, for inaugural speech. Welcome, sir. Principal, sir. Welcome, sir.
Hello, sir.
This is your session, sir.
Okay, thank you, ma'am. Welcome, everybody. Welcome, Adventure, sir. Are you here? Am I listening? Am I audible?
You are audible, sir. You are audible, sir, but video is not on.
Video is not there.
Video is not on, sir. Your video is off, sir.
Okay.
No, okay, sir. Actually, ma'am, I think there is any problem. That's why I can't see.
Okay. Welcome, everybody, in this webinar. We initially started or fixed another name. Actually, theme was like that. Every day we struggle with the corona epidemic situation, and in this state, what actually do and don'ts, it is not clear for everybody. We want to inform our students, our colleagues, and of course to the guardians, what actually to do scientifically to avoid from this epidemic situation, whatever may be. Madam select very perfect topics: a voyage across the COVID-19 pandemic era, physiological, pathological, and psychological fears and facts. And in this webinar, I like to welcome everybody, especially at our Sabang Sajanikanta Mahavidyalaya. Sir, especially I feel proud in this very occasion to announce this international webinar organized by the Department of Physiology in our Mahavidyalaya in collaboration with the Internal Quality Assurance Cell. I feel proud to warm welcome our honorable distinguished speaker and resource persons. We set actually 2 days in 2 different sessions. Day 1 on today, there Professor Somnath Roy, former professor and HOD, Department of Human Physiology and Community Health with the Sabang University, and Edmund Chua, company Sci-Tech Bioscience USA. He will talking about some spectral flow cytometry and COVID-19. It is very, as per my best of knowledge, the cytometry is a very advanced method to detect the cellular activity, cellular position, cellular details, subcellular details like that. He will say about day 2 on 2nd, 2nd, 9th and 20, Professor Samuel Roy, Professor and Dean, National Institute of Pharmaceutical Education and Research, Kolkata. He is actually Chief Scientist, CSIR, Indian Institute of Chemical Biology, Kolkata, and former Vice Chancellor, Honorable Vice Chancellor, Coach Bihar Panchanan Burma University. And interestingly, and this is our main, another addition. A team of distinguished doctors. A team of distinguished doctor of infectious diseases and Beleghata Hospital. Actually, its pet name is ID and BG Hospital, Kolkata. They will discuss about the fears and facts regarding the COVID-19 pandemic. Sir, I am also expressing my warmth and deepest gratitude to all in this webinar and waiting for your expressive and your thought-provoking realizations over the issues. In this webinar, I am also welcome to all the participants being present here from the different corners and also from abroad. I must congratulate to organize to select these relevant topics to our organizing secretary, Dr. Jhumu Manna, Assistant Professor and Head, Department of Physiology. And I am also welcome to our team of technical committee, subcommittee. There are 6 very young teachers, Mr. Sumanto Chaudhary, Supriya Konai, Mr. Somnath Roy, Atanu Samanta, Monalisa Banerjee, Rajdeep Dev, and others also. They are actively present here, and they looking total matters, look after the total matters of the technical supports. I would like to welcome Dr. Salim Khisti, Associate Professor, Economics and Department of Economics, and he is also coordinator of the IQAC. Members of the advisory subcommittees, Abhik Mukhopadhyay, Dr. Abhik Mukhopadhyay, Associate Professors of Commerce, and Shunita Mitra Sarkar, Assistant Professors, Political Science, for their valuable support. In these connections, I eagerly waiting for your valuable talk, Advan sir and Professor Somnath Roy. You are most welcome, sir. Before concluding, please mute your microphone, and if you want to say, of course, you are most welcome when in the discussion or interaction sessions you will be asked for, and you may discuss with our distinguished professors and teachers. Thank you. Sir, কেমন আছেন? সোমনাথ স্যার।
ভালো আছি, তুমি কেমন আছো?
আমি ভালো আছি স্যার। সোমনাথ স্যার, he is my teacher also. I learned from him about some immunological facts. স্যার নিশ্চয়ই আপনি অ্যাক্টিভলি, তিনি সোমনাথ বাবু স্যার অ্যাকচুয়ালি প্রফেসর সোমনাথ রয় 2 দশক ধরে অ্যাক্টিভলি ইমিউনোলজির উপরে কাজ করছেন, এবং তার সম্বন্ধে অবশ্যই বলবেন আমাদের ঝুমু ম্যাডাম, তার ডিটেলস নিশ্চয়ই দেবেন। তবে স্যারকে দেখে অনেকদিন পর দেখলাম, অনেকদিন আমার ইউনিভার্সিটি যাওয়া হয়নি, আপনার শরীর একটু ভেঙে গেছে দেখে মনে হলো, একটু তবে রোগা থাকা ভালো স্যার।
Thank you, Tapun, for your address, introduction and everything. আমি যখন নাম শুনলাম ঝুমুর কাছে, তোমার কাছে, I am really delighted যে আমাদের ছাত্ররা একটা কলেজের ইতিহাস প্রিন্সিপাল হিসেবে চালাচ্ছে। That is very encouraging for us, especially for the teachers.
ভাই আমি আমার কাছে ফরচুনেট এটা আপনাদেরকে টিচার হিসেবে পেয়েছি, যতটুকু পেরেছি শিখেছি। থ্যাংক ইউ স্যার, থ্যাংক ইউ।
Thank you.
Oh, madam, please you take yours and introduce our thank you.
Thank you, sir. Thank you, our honorable patron and principal, Dr. Tapan Datto, for our nice welcome to our speakers. Now it is the session of lecture. At first, our first speaker is Professor Somnath Rai. May we recall Sir Omul from Dagghor, written by Rabindranath Tagore. That child with the incurable disease had to live in a room. At first, this imprisonment seemed too unbearable to him. But from the day he knew the king's letter would come in his name, there is no more sorrow in his waiting. We are also waiting for the king's letter, that is corona vaccine. In fact, only science can show us the way. Our first speaker, Professor Somnath Rai, will give the idea of the king's letter, that is corona vaccine. The title of its topic is Structure, Pathogenicity and Vaccine Development of COVID-19. Professor Rai is the founder of Master Degree or PG course in Vidyasagar University. He obtained his PhD and MSc degree from Calcutta University. His major scientific contribution is drug-resistant microorganisms like superbugs and Plasmodium falciparum, malaria. Actually, he has the contribution on country's malaria control program. He has 20 PhD scholars. They have completed their doctoral degree. He has published several research papers, among which 114 are international and 19 are national in peer-reviewed journal. Professor Rai is also author of several books and book chapters. His popular book is Basic Concepts of Immunology. He is also the editor of the prestigious journal, Advanced Techniques in Clinical Microbiology. Professor Rai has elected as the president of Section Medical Science, including Physiology, of the Indian Science Congress Association for the year 2016 and 17. He has been elected as Fellow of West Bengal Academy of Science of Technology from Ministry of Science and Technology, Government of India in 1993, and worked with Professor Jyotirmoy Das, Director of IICB, in a project. Professor Rai received several awards, including Dr. P.K. Devnath Memorial Award in 2015, from the Society of Ethnopharmacology, Professor B.B. Sarkar Memorial Oration Award in 1996, and Srimati Shakuntala Dasgupta Memorial Oration Award 2009 from Physiological Society of India. I hope the session will be interesting. Welcome, sir. This session is now yours.
Thank you, Jumu, for your nice introductions. And it is my pleasure to join this seminar. I'm grateful to Sabang Sajanikanta Mahavidyalaya, especially the principal, Dr. Tapan Kumar Datto, and head of the department, Dr. Jumu Manna and Monalisha, for giving me the opportunity to share some presentation. Today, I will talk on the title of my lecture is Structure, Pathogenicity and Vaccine Development of COVID-19. Can you hear me? Jumu?
Yes, sir. Yes,
Yes,
sir.
sir.
You're audible, sir.
You
Okay,
are
Continue.
Sir,
audible, sir.
okay,
everything all right, sir. Everything.
okay, nice. So this COVID-19 or coronavirus was first found in Wuhan city in Hubei province, China, and it is highly transmittable, infectious, and pathogenic, and responsible for the disease severe acute respiratory syndrome. Shortly, it is termed as SARS. Later, it quickly spread around the world. On January 30 in this year, World Health Organization declared this outbreak as pandemic across the continents. It originated on December 2019 from the market, possibly from the bat. Later, it was transmitted in different primates like civet cat in case of SARS-CoV in the year 2002 and possibly in pangolin in case of SARS-CoV-2 in the year 2019. Though there are still some controversies regarding the transmission route for SARS-CoV-2. In lower right side, there are certain strains causes upper respiratory tract infection and showed mild symptom of the patients. Again, on the other strains like SARS-CoV, MERS-CoV, and SARS-CoV-2 causes or responsible for the lower respiratory tract infection, which causes severe pneumonia. This table showed comparative incubation period, recovery days, and reproductive number. We found the long incubation period of COVID-19 virus, that is 2 to 14 days. Lower table shows infected cases of death of SARS and MERS. Now the pathogenesis of COVID-19, we already know the symptoms of coronavirus, that is fever, dry cough, and shortness of breathing. And in severe cases, we found acute pneumonia, acute respiratory distress syndrome. In case of more critical cases, septic shock, respiratory failure, multi-organ failure, and about 15% of COVID patients develop pneumonia, and about 5% of them go on to have multi-organ failure. In the left side, there is some schematic structure of the coronavirus. It has genomic RNA, which is covered by an envelope, and spike proteins are arranged in the outer part of the envelope. And these spike proteins are very interesting. And we'll talk in our later side that most of the vaccines and scientists are trying to use these spike proteins for the development of the vaccine. So it has two parts. Spike has two parts, S1, S2, and these spike proteins usually bind with the receptor, which is known as ACE2 receptor. The full name of this receptor is angiotensin converting enzyme 2 receptor, which was found in the lungs of the human host. So this is the detailed structure of the coronavirus, and we have found that coronavirus genome size 30 kb, positive RNA genome, and COVID particles are pleomorphic with a helical nucleocapsid, and it encodes 4 major structural proteins like spikes, envelope, membrane, and nucleocapsid. Spike protein facilitate entry into the target cell. These spike proteins are used by various scientists and doctors for vaccine development to produce antibody of human host. This is the three-dimensional structure by the crystallography, protein crystallography of the coronavirus. And these spike proteins plays a very important role, which I have mentioned earlier, for the viral entry. And this is this portion is, this upper part is known as upper part is receptor binding domain, and it is the most variable. The amino acids are most variable. This is a very interesting report and part of the coronavirus genome. And the lower part is fusion machinery and is more conservative on contrary. So upper part is variable, but the lower part is conservative and helps in membrane fusion and deposition of nuclear capsid. This spike protein virus greatly, as these more stickier spikes lead to cytokine production, which I will aim to later slide. And these spike proteins can be useful to make vaccine preventing COVID-19, as scientists suggest. And this is the genetic structure of COVID-19 virus. It possesses 14 open reading frame and which encode 27 proteins. This means though eukaryotic translations are generally monocystronic in nature, this virus encodes 27 various messages from a single RNA. And as we know, the mRNA actually responsible for the synthesis of the protein. So these 27 various messages from single RNA makes it very dangerous. And actually, Moderna Biotech in US take these advantages to use this one portion of the mRNA, which is responsible for the synthesis of the spike, producing spikes protein. As the sequencing of this genome already published in reputed scientific journal, so this is more easier to this Moderna Biotech to initiate to prepare the vaccine. And this 5 to PP1AB portion are termed as a non-structural protein, and the rest are structural and accessory proteins. So these are the different structure of the coronavirus. So how it entered into the human lung, or what are the route of entry? And you have seen in the structure the mouth or nose. So the main target one, the question arises, why should we use the mask? The answer is to prevent the virus from this entry point, that is mouth or nose. So the main target, or because the main target organ of this deadly virus is lungs. So the virus particles enters into the body through the nose or mouth, and this is shown in the picture. Respiration carries some of the particles to the lower respiratory tract, where these spike proteins acting like a lock and key into the epithelial cells, as well as in the air sacs of the lungs. So in the side picture, right side picture, we have seen that the spike and the receptor, this is the lock and key model. So after a few days, if these spike proteins are unlocking with the receptor of the lungs, this virus, after hijacking the cell's machinery, this means virus enter into the host cells and hijacking control the host cell machinery for its replication, multiplication, also infecting the adjoining cells. Immune system has here an important role. If this immune system unable to fight back, the virus goes down the windpipe and infect the lungs. In the next two slides, we can compare the healthy lung structure, changes of the lung structure. In the next slide, we also find what are the difference between the normal, moderate, and severe patients. So this is the healthy air sacs of the lungs covered by capillary vessel, and as a physiological student, we know the exchange of oxygen and carbon dioxide in the picture, that is in the red arrow, where carbon dioxide from capillary vessels to the lungs goes to the lungs, and from lungs, from the alveolus, from the air sacs, oxygens come to the cells and bind with the hemoglobin of the capillary vessels, and that is distributed to the different cells. And nutrition is also carried out as supplying the nutrition as
Just a
well as
minute, sir.
oxygen.
Sir,
Yes.
hello.
Yes,
Sir,
yes.
you are not presenting your slides, sir. There is an option on the left, rightmost side, present now. You have to click there.
তোমরা কি দেখতে পাচ্ছ না?
না স্যার, স্লাইড
No,
দেখা যাচ্ছে না।
সাই দেখতে পাওনি?
actually, sir, you have to present that, sir.
Present now, there is an option present now. Rightmost side,
আগের
sir.
স্লাইড গুলো দেখতে পাওনি? আগের স্লাইড গুলো
No, sir.
কোনো স্লাইড দেখতে পাওনি?
No, sir.
তাহলে সেগুলো তো তখনই বলবে, স্ট্রাকচার গুলো যখন বলে যাচ্ছি তখনই তো আমি কারেক্ট করে নিতাম। দেখা যাচ্ছে? এবার দেখা যাচ্ছে? দেখা গেলে বলবে।
স্যার মনে হয় প্রেজেন্ট করতে পারছেন কোনোভাবে। স্যার হ্যালো,
হ্যাঁ, এক সেকেন্ড দাঁড়াও, আমি চেষ্টা করছি। এবার দেখা যাচ্ছে? হ্যালো?
no sir, sir, you have to present your screen, sir. Yes sir, yes sir, now it's coming.
আচ্ছা, আগের কোনো ছবি দেখতে পাওনি?
না স্যার,
একবার
আগে তো দেখা
স্লাইড
যায়নি।
একটু পাস করিয়ে দিন স্যার।
হ্যাঁ, আমি তাহলে ছবিগুলো আরেকবার একটু দেখিয়ে দিচ্ছি।
জাস্ট একবার পাস করিয়ে দিন।
So this is the structure of the coronavirus, and it has genome size 30 kb, RNA genome 125 nanometer diameter, and it encodes 4 major structural protein like spikes, envelope, membrane, and nucleocapsid. And these spike proteins actually facilitate the entry into the target cell of the host. Next. And this is the 3-dimensional structure of COVID-19. Have you seen whether it is visible? Remove?
Yes, sir. Yes, sir. Visible.
Visible. Okay. So this is the 3-dimensional structure of COVID-19, and this is the upper part of the structure. This is the protein crystallography structure. This is the upper part. And this upper part is receptor binding domain, and it is the most variable part. And then this is the lower part. This is the fusion machinery and more conservative. So upper part is variable and lower part is conservative. And these spikes makes the virus deadly, as this is more stickier spikes, and which ultimately leads to more cytokine production and damaging the inner vital organ of the host. So this is the genetic structure. It possesses 14 open reading frame encoding 27 proteins. Though eukaryotic translation are generally monocistronic in nature, this virus can encode 27 various messages from a single RNA, which makes it very dangerous. Actually, Moderna Biotech used this opportunity to use 1% of this mRNA to prepare the vaccine, as the sequencing of the genome already published in the reported journal. And the 5' to pp1b portion are non-structural protein, and the rest are structural protein. So this is the route of entry of the human lung. Why we should use the mask? As the virus entered in this route, so by using mask, we are trying to prevent the virus. So this is the route of entry of the virus. So the main target organ of this deadly virus is lungs. The virus particle enters the body through the nose or mouth, which is shown in the picture. Respiration carries some of these particles to the lower respiratory tract, where these spike proteins acting like lock and key. In this right figure, we have found the spikes and the receptor. This is known as a lock and key model into the epithelial cells as well as in the air sacs of the lungs. After a few days, these spike proteins are unlocking with the receptor of the lungs, and the virus, after hijacking the cell's machinery, because it will infect or enter into the normal cells of the human host and replicates and multiply and also infecting the adjoining cells. So immune system has here an important role. If the immune system unable to fight back, then virus goes down to the windpipe and infect lungs. So in the next two slides, we can compare the healthy lung structure and changes in the lung structure of moderate patient and severe COVID patient. So this is the normal structure of the lung of the human being, and this is the in the left side they are the air sacs of the alveolus and the capillary covered by these air sacs. And we have seen here the exchange of oxygen and carbon dioxide. As a physiological students, everybody knows that carbon dioxide goes from the carboxyhemoglobin to the lungs for the expiration, and oxygen come from the lung to the vessels and bind with the hemoglobin portions, and that will be distributed to the different cells of the body. So alveolus consists of type 1 and type 2 cells. And after infection, type 2 cells release inflammatory cytokines to recruit macrophages. Now question arises, what is macrophage? As a physiology student, you already know the 5 types of WBC. We have seen here the main 3 WBC are responsible for the COVID patients for the interaction, like neutrophil, lymphocyte, and monocytes. So these monocytes, when entered into the blood, it is known as macrophage. This is the structure of the macrophage. So role of macrophage here is to release cytokines. Macrophage has several functions like antigen presentation and another function, but here it causes the release of cytokines, and these cytokines are responsible for the vasodilatation and allow more cell to reach the injury sites. Especially neutrophil also accumulates in the injury site and release reactive oxygen species. And as you know, the biological student, there are 3 to 4 types of reactive oxygen species like superoxide ion, hydrogen peroxide, hydroxyl radical, and these radicals actually try to destroy the virus infected cell. But if that is not properly possible, and inflammation ensues, and if the inflammation in severe condition, the protein-rich fluid enters into the blood, causes systemic respiratory syndrome, which is known as SIDS, and causes septic shock and multi-organ failure. In the next structure, we have found the moderate and severe patient lung structure. And this is the gross anatomy of the lungs. In the left side, 2 structure of the air sacs of moderate patient's lung, and next severe patient lungs. We have clearly found that the fluid accumulates in the moderate COVID patients in alveoli, and fluid dilutes surfactant and triggering the collapse of air sacs and decrease the gas exchange. In severe patient, you see in here that the color of the yellow, just yellow-like color, when it fools the air sacs. So in severe patient, protein-rich fluid accumulates full air sacs. Neutrophils and macrophage accumulates. These are the neutrophils, some of the neutrophil structure and macrophage structures accumulate and releases cytokines and causes collapse. You have seen some collapse starts. collapse of air sacs as well as alveolus, causes damages of type 1, type 2 cells, causing acute respiratory distress syndrome, and neutrophil macrophages accumulates and releases cytokines. In the next slide, we can correlate the immune system and cytokine. Now, what is the immune system? We have a defensive mechanism which is known as immune system. It is just like the defensive mechanism of a country. And white blood cells of our body can protect us from any foreign attack like virus, bacteria, and toxin. These WBCs are responsible for secreting some of the bullets to destroy the microbes. These bullets are known as proteins or cytokines. So I have named this site as a cytokine storm because this is the previous indication or primary indication symptoms of findings. This is the probably previous symptoms or findings before the death of the severe COVID patients. Cytokine tremendously, the concentration of the cytokines are tremendously increased. So these cytokines are immunoregulatory proteins. According to the scientists and doctors, the concentration of these proteins and cytokines are tremendously increased, tremendously. But in physiological condition, these cytokines are responsible for the giving the different signals for the physiological function. But instead of doing good, these cytokines causes the bad or destruction to the body. T cell, B cell, macrophages, and other cell can secrete the different type of cytokines. And when these cytokines are tremendously increased, which is termed as a cytokine storm or cytokine surge, interleukin 6 and interleukin 1 levels are tremendously increased. And in more critical cases, septic shock, respiratory failure, and multi-organ failure, and ultimately death of the patient occurs. So before going to the vaccine development, we discussed the important discoveries of Edward Jenner and Louis Pasteur, that is history of vaccine development. Thucydides in the year 430 BC in Athens first used the term immunity to an infectious disease. He was an historian. He had some power of observation like scientists. Though he was historian, but he had some power of observation like scientists. He wrote in his book, that is 430 BC, can you imagine? Only those who has recovered from plague could nurse the sick, could nurse the patient, could nurse the plague patient. Why? Because they would not contract the disease again. So the recovered patients acquired immunity against the disease. Edward Jenner, an English physician, was the first person to scientifically perform his manipulations under controlled conditions and to record his experimental observations and conclusion to achieve a dramatic success, ushering in the dawn of immunology. A dreadful disease, smallpox, was found at that time. Surprisingly, he observed the milkmaids who had recovered from cowpox never suffered from smallpox, a dreadful variety of disease. He took the materials from the cowpox puzzle and injected into the only 8 years old boy named James Phipps. Can you imagine? In that time, he took this risk. Thereafter, the boy was inoculated intentionally. The boy was inoculated intentionally with smallpox virus. Surprisingly, he found the boy did not contract or develop smallpox. That is in the year of 1798. This method known as Jenner's technique, and this is the groundbreaking and pioneering work of Edward Jenner. And this pioneer work published in all newspapers and spread around the world. Next, the Louis Pasteur discoveries. He was a French scientist and was a professor of Lille University in France. He had been working on fowl cholera or chicken cholera. After coming back from summer vacation, he injected chicken or fowl cholera again to the experimental subject. He observed instead of death, chicken were survived. Now he thought, what are the reason? He surmised that he thought disease was due to the low virulence power of the bacteria. And to confirm his idea, he introduced heat-killed fowl cholera. Treat this cholera, fowl cholera, by heating, that is known as heat-killed fowl bacteria, and what is later it was termed as attenuated bacteria or attenuated strain. So he introduced heat-killed fowl cholera to the chicken and found the same observation like previous experiments. He used the term as attenuated bacteria to this fowl cholera, and to honor the Edward Jenner, he named this attenuated strain as vaccine, because in Latin word vacca meaning cow. Pasteur concluded that aging also reducing the virulency of the bacteria. In the year 1885, Louis Pasteur produced a vaccine for rabies. So how the vaccine are prepared and what are their principles or methods to prepare the desired vaccine? There are several methods for vaccine development. Major types are killed vaccine, live attenuated vaccine, subunit vaccine, toxoid vaccine, conjugate vaccine, and recombinant or genetically engineered vaccine. Oxford University vaccine was manufactured by the application of recombinant DNA techniques, commonly known as genetic engineering. Oxford vaccine is from United Kingdom, and team leader of this group, Sarah Gilbert, she is Sarah Gilbert. So they are using a vector which is known as adenovirus vector, and currently being prepared by Oxford University and Jenner, according to the Edward Jenner, the institute was named. So the Jenner Research Institute at Oxford University introduced this Oxford vaccine, and Sarah Gilbert is the team leader. So the simian adenovirus vector, which is genetically engineered virus that causes the common cold in chimpanzees. Previously it was known as ChAdOx1, but now it is called as AZD1222, containing full length of structural surface glycoprotein protein or spike proteins of SARS-CoV-2 with a tissue plasminogen activator leader sequence. According to the Lancet July 20 publication, ChAdOx1 showed an acceptable safety profile for boosting and increase the both humoral as well as cellular response. So this is the Russian vaccine Sputnik V, and Russia on August 11 declared that it had become the first country to approve a vaccine against the novel coronavirus. The Sputnik V vaccine will involve two jobs using two different vectors of human adenovirus. As this vector had a significant track record of use in previous vaccine, it had more historic data and supporting its safety than some other potential COVID vaccine. Some people may develop very strong protective immunity while others may not have light symptoms. In the phase 3 trial, they will try to involve 40,000 people, and the TASS news were openly reported earlier. The vaccine was developed by Gamaleya Research Institute and the Russian Defense Ministry. Russian President Vladimir Putin claimed that one of his daughters inoculated the vaccine and she was fair, good, and though he has passed all the necessary tests. Now come to the Moderna's mRNA vaccine, USA, and Moderna uses genetic messenger RNA to make the vaccine, which I have already presented you. And researchers at Moderna and the National Institute of Allergy and Infectious Diseases, USA, identified the part of the sequence of that encodes a spike-like protein on the surface of the virus that attaches to human cell, helping the virus to infect them. The idea behind the Moderna vaccine is to inject the mRNA for part of the spike protein and have it slip into the cell of a healthy person, which is then follow its instruction and crank out the viral protein. That protein act as a red flag, because if you are introduce the mRNA, then mRNA will be responsible for synthesis of the protein, that is spike protein, because the spike mRNA they are using. So that protein acts as a red flag for the immune system, stimulating it to produce antibodies, and that will prevent infection by blocking the action of the spike if the person is exposed to the virus. So this new technology for genetic immunization are rapid and produce a product that is highly potent and producing immune responses. The scientist Dr. Muligian said, today's RNA result confirmed that there is a grand potential. That is next Indian vaccine, that is Covaxin, Zycov-D. India has two indigenous vaccine candidate, Covaxin by Bharat Biotech and Zycov-D by Zydus Cadila. Both these two companies are nearing completion of the phase 2 trials. Covaxin is also collaborated by the Indian Council of Medical Research and Virus Institute, that is National Research Institute of Virology, that is NIV Pune, with Bharat Biotech. Serum Institute of India, which is also in Pune, is all set to start the phase 2 trial of COVID-19 vaccine developed by University of Oxford and AstraZeneca. British Swedish firm has partnered with Serum Institute of India to manufacture Oxford COVID-19 vaccine for India and other low and middle income countries. First dose will be given on day 1 and second dose will be given schedule on 29 days. So there is a 4 weeks gap. Now to conclude, we sincerely hope that out of so many different avenues being explored, we'll find a sustainable solution to fight this pandemic. Till that moment, we have to keep our faith on our scientists, doctors, and medical system, because history teaches us to do the same. Alongside, we have to abide by all the rules, regulation, health advices provided by the world experts for a long time period. As general public, this will be our role in fighting this pandemic together with everyone. We hope very soon we'll defeat this dark situation to arrive at a vibrant virus-free world. Let us dream of such a world together. So one of the Nobel laureate, Professor Taku Honojo, told that this is a time for all of us to work together to fight this common enemy. We cannot delay our moment in this effort to save the lives of our fellow humans. Thank you very much for patience hearing. Thank you.
Thank you, sir, for a nice speech. We are a little bit late, sir. Please pardon us. Thank you, sir. Very happy to see you for a long gap, sir. Monalisha, is there any question?
Single
Hello?
question may be asked.
No, ma'am, there is no question in the chat box, but participant can ask direct questions. We can enter 1 or 2 questions.
A single question, please. Sir,
Yes.
Professor Rai. Sir, we are a little bit late and Mr. Edmund Chua is waiting. Uh,
Okay.
He's
Okay.
maintaining our
Okay.
ISP. Sir, we
Okay.
will
Okay.
discuss later, sir, after which session.
Okay. Okay. No problem.
Will you please,
No
sir?
problem. Okay.
Thank you, sir.
Okay. Thank you.
Sir, please be with us.
Sir, now we are going for the next phase of our lecture session. Welcome Mr. Edmond Chua from SciTech Biosciences, USA. Welcome, sir.
Yes,
After
hello,
the-
hi.
Yes, sir, you are audible, sir. Welcome in our digital platform. We are very happy to get you. Sir, after graduating from Nanyang Technological University, Singapore, uh, Edmond was hired to manage A-Star IMCB's flow cytometry core facility. As a core manager, he played an important role in SOP optimization, developing and validating high-parameter flow panels. His previous work has also fostered close collaboration with industry players and has managed several human clinical trials and non-human primate research projects. In his role as the application specialist for Asia Pacific, Edmond is a familiar face in the flow cytometry community around the region. He has conducted several workshops and presented seminars on spectral flow cytometry at multiple conferences. In his new role as SciTech's Sales and Marketing Manager East Asia, he has been actively pursuing contacts and collaboration with the APAC flow cytometry community. In his free time, he enjoys playing badminton. Sir, we are lucky enough you are with us. Please continue, sir.
All right, thank you for that kind introduction. So yes, my name is Edmund. I'm the Sales and Marketing Manager of East Asia for SciTech. We are a full spectrum flow cytometer solutions company. So we have been around for about 25 years. But only in the last 5 years have we launched our own full spectrum flow cytometry, which is very different from conventional flow cytometry. And today the topic that I'll be talking about is full spectrum flow cytometry and how it has its application in COVID-19 research. So being able to do full spectrum flow cytometry enable our users to go beyond 30 colors to be able to do 35, 40 colors in a single cell. So they can actually look within a single cell, 40 different markers. And that has helped everyone or anyone that is working on COVID-19 research to be able to develop, diagnostic tools or any way to assess vaccine response, be able to assess severity of the disease of COVID-19 just using their blood or their PBMC. All right. So a little bit of outline of what we're going to go through today is that's the introduction to full spectrum flow cytometry, how different it is from conventional flow. We are also going to talk about fluorochrome signatures because we are no longer going to identify our fluorochromes or our colors by a single channel, but we are identifying them by their signature. And every fluorochrome kind of have their own unique signature or kind of like a fingerprint. We'll talk about some of the performance characteristics of our instrument, what are the advantages of spectral cytometry, why people are moving to spectral cytometry and moving away from conventional cytometry. And we'll also share with you one of the ways that we enable our users to simplify multi-parametric flow. So as you increase the number of colors in your panel or in your experiment, a lot of times it becomes very complicated and it becomes very difficult. And how we can help in simplifying that procedure is by introducing something we call the reference control library. So the reference control library is something that we store the reference control in the software, and so you don't have to prepare single stain controls every single time that you're running the same experiment. So you can store it in there and you can pull it out anytime you want to run the same experiment again. We'll also have a comparison of our instrument against other conventional cytometers. And finally, we'll show some of the COVID-19 data that we have generated so far in collaboration with a lot of labs around the world that are working on COVID-19. So before we move into what full spectrum flow cytometry is, we have to revisit how flow cytometry has evolved in the last few decades. So it started off in the 1980s with one laser, two color system. FIT-C and PE were the first two dyes that were used in conventional flow cytometry. And then, 10 years later, there was an introduction of a new laser, the red laser, with additional APC and Per-CP dyes that were introduced. And then we have fast forward that to like 2020, 2015, where we have 5 laser systems that are out in the market now, and many different colors and channels available. And what you want to notice is that throughout the last few decades, it has always been the same way of detecting a fluorochrome or detecting a signal, by using a very narrow bandwidth of light to identify the fluorochrome or to identify the peak of the fluorochrome signal. And we are missing out all of these other signals that are outside of the detection. And so it is using one detector at detecting at the peak of the fluorochrome to identify the fluorochrome. And so how is spectral cytometry different from conventional cytometry? Spectral cytometry is termed as using dispersive optics, such as prisms or gratings, where we disperse this collected light across a detector array, and that allows the full spectrum from each particle to be measured. So as you can see over here, we are not only just looking at the peak of the emission of the fluorochrome to identify it, but we are actually taking the entire spectrum or the entire signal, and we take the pattern of how the signal is shown on the instrument as a signature to identify the fluorochrome. So it is not any different from a conventional cytometer, but only that the detection method is different. We still have lasers in a spectral cytometer with cells labeled with your antibodies, fluorochrome. The only difference is that we have our dispersing element and multi-channel light detector that is different, or we are changing the way we are detecting our fluorochromes. But in terms of laser, fluidics, it's all very, very similar with conventional systems. So I'm going to introduce to you our 5 laser system, which is our most advanced system that a lot of our users are using around the world for COVID-19 work, because the 5 laser system enable the users to go beyond 30 colors. We have been able to show that we are able to go up to 40 colors in one tube, in one sample, to be able to run all 40 markers in the same time, at the same time. So over here we have our 5 laser system, where we have the UV laser, the yellow-green, the blue, the red, and the violet laser. Each of them have its own individual laser line, and then they are spatially separated into the flow cell to excite your fluorochromes. And the emission signal is then collected into each of the detector modules here. Each laser has its own detector module to detect the entire emission spectrum after excitation by the laser. And then we also have 2 side scatter detectors here, one of the blue laser and one of the violet. The violet laser is good for detecting really, really small particles like exosomes, viruses, any nanoparticle delivery that you are using for vaccine work. It is also can be detected using the violet side scatter detector, right? So We talked about the instruments layout, and we're gonna show you the filter configuration. So I've showed you the 5 laser systems, but we have different configurations. We have 1 laser systems all the way up to 5 lasers. So for the 1 laser system, because we talked about a full spectrum, we have a total of 14 channels or 14 detectors in the blue laser 1 laser system, and we are collecting the entire emission wavelength starting from the point of excitation. Because it's a blue laser, it starts at about 488 nanometer. We start very close to that, and then collecting the entire wavelength of light until 830 nanometer. So as you can see over here, we are collecting with very minimal wavelength of light, about 15 to about 30 nanometer wavelength of light for each of the detectors. And we are collecting the full spectrum from 498 all the way to 830 nanometer. And for the 2 laser, it's the same thing. We are collecting from near the point of excitation for the red laser, which is 640, all the way to about 830 nanometer. And so it only has 8 detectors because it starts off at a later wavelength. 3 laser systems, the addition of a violet laser. We have 16 channels for the violet laser. The violet laser is a 405 nanometer laser, and we are collecting from about 420 nanometer all the way to 830 nanometer. So you will want to notice that again, we are collecting the entire emission spectrum with no gaps in between. For example, V1, 420 to 435, V2 starts at 436 to 451, so on and so forth, all the way to 830 nanometer, so that we can collect the entire emission spectrum. 4 laser systems has additional yellow-green laser, and so again, additional 10 yellow-green channels here to collect the emission spectrum from 567 to 830 nanometer. And finally, with the ultraviolet laser, we have another 16 channels here collecting from near the point of excitation for the UV laser all the way to about 830 nanometers. So for our 5 laser systems, we actually have a total of 64 different detectors on the system. And we have been able to show that we can do 40 colors on a 5 laser system. Of our 1 laser system, we have shown that we can do 9 colors on just the blue laser alone. We can do 9 colors on the blue laser alone. So what do we actually do with all of these 64 detectors that we have in our system? It's very important because we're collecting all of the data from these detectors here, so to form this full spectrum signature. So over here in the x-axis, you will see the channels or the detectors in the filter configurations that we have seen just now. And in the y-axis is our MFI, or median fluorescent intensity. So we are actually using the entire emission spectrum of the UV laser collected here, of the violet laser as well, the blue, the yellow, green, and the red laser, and then stitching them together to form this spectral signature. So we are now identifying a fluorochrome by its signature by looking at how the patterns of these spectral plots would look like to differentiate between different fluorochromes. So every signature or every fluorochrome will have its own unique signature. And so we can, based on all of these different channels, to find the differences and determine, whether it is one or the other fluorochrome. So this is an animation to show you how we actually generate the full spectrum signature. So again, we are collecting the entire emission spectrum from the point of excitation to about 830 nanometer. And these are all the individual spectral APD modules that are collecting the emission spectrum from each of the lasers. And you can see that we are then stitching them together after we have collected the information. And that gives us the spectral signature or spectral plot of how a fluorochrome looks like. We are then using this mathematical algorithm called spectral unmixing, also known as ordinary least squares, and we are calculating or determining if your multi-color sample, which has all your 40 colors or 35 colors, has this particular signature in it to identify whether this fluorochrome is in the sample. All right. So we have actually made it easier for our users by doing finding all the fluorochromes that are commercially available and actually put down their signatures so that you can then easily see how the signatures look like. So we have some very unique ultraviolet unique signatures like here. So all of the BUV dyes, the brilliant ultraviolet dyes, are unique and they can be used in a combination in one panel. The violet signatures here, for example, you can see that each individual spectral plot here or signature here has a name, and that would mean that this has a unique signature. If you see that the plot has multiple names, it means that you have to pick or choose only one of these dyes to use because their signatures are very, very similar. So it would be very difficult for the software to pull apart the differences. So what you want to notice is that things like BV421 can be used in combination with PAC blue because they have their own separate spectral plot and they are very different in terms of their signature, if you can see over here. All right, in this region, these two dyes are very different. So you actually can use these two dyes in combination. Another very interesting combination is the BV605 in combination with Q.605, which you would not be able to do in a conventional cytometry system. It would not be able to pull apart these two dyes. The blue laser unique signatures are over here, where you can see we are able to pull out like BB515 and FITC. We can also separate out GFP from FITC or GFP from AF488 because they are very different signature as well. So fluorescent proteins have their own signature, which looks different from what FITC looks like or PE looks like, giving you the advantage of being able to use more colors in combination. We also have things like, PE Sci-Fi, PE Sci 5.5, PE Sci 7, PE Alexa Fluor 700, which have very unique signatures and can be used in combination. And finally, we come to the red laser, where we have APC and AF647, my favorite example of a highly overlapping dye, where you actually can see that in a conventional system, APC and AF647 would not be able to use together in a panel or in an experiment, but you can in a spectral analyzer or spectral system because you are looking at the full spectrum of the dye, and you can see very clearly that there are differences in both of these signatures. So it sounds like it's a very complicated workflow on Cytec systems when you're trying to do your flow cytometry experiments, but I can tell you that they are as straightforward or as the same as what you would do in a conventional system. You would run your unstained control, which means that your cells with nothing in there, with only just the cells that have been treated the same way that you have, and then running your individual single stain control to identify the signature of each of the dyes that you have in your experiment. And then unmix, which is equivalent to a compensation step in a conventional cytometer. It's an automatic process that will do all the calculation in the background. And then you run your multicolor sample in order to see in real time how the sample looks like when you have your multicolor sample. So it will come out in your FACS plot for you to draw your gates. So performance characteristics, I'm going to show you some of the performance of how and why spectral cytometers are very important when it comes to flow cytometry. We are using avalanche photodiodes, so different from conventional systems that are using PMT-based systems or photon multiplier tube-based systems. Avalanche photodiodes are smaller and more compact, which gives us a lot of space saving. So our 5 laser system can actually be placed on the bench. It is very, very small because we are able to save the space on our detectors. As you can see over here, this is the size of our lid to cover all of our detectors, where we can store up to 50 channels, as compared to a 3 PMT trigon module here in a conventional system. The PMTs are very large and they take up a lot of space, and so it is often not very economical when it comes to space and also pricing. So another thing here we have is why we have high sensitivity APDs, and how we quantify detectors is usually by looking at their quantum efficiency, which is what we term as the ability to convert photons into electrons. So when you look at the graph over here on the right, you can see that the PMTs in the red line here, the wavelength on the x-axis and the y-axis being quantum efficiency. PMTs on the red line here starts off at about 20% quantum efficiency. And as it goes off into the higher wavelength, it actually drops to almost 0.1%. So it is a huge drop in terms of quantum efficiency as you increase the wavelength of light, meaning that things like your APC size 7 will need a lot more signal in order to convert that into electrons, in order to get the signal on your instrument. So when we look at APDs in the green line here, you can see that already at the start, APDs are performing at double, almost 40% of quantum efficiency of a PMT, and it even goes up to about 100% as you hit the higher wavelengths. So the APDs actually have a higher quantum efficiency across a broader range of wavelengths as compared to the PMTs. And the low electronic noise of the system that we have measured using hard dye beads, where we look at the fluorescence signal to the APDs by blocking the signal, and then we look at the RSD of the noise by increasing the gains. So because we are using APD systems, they are known as gains, and you can see over here, x-axis over here with gain, and y-axis is RSD, even increasing to a high number like 3000. Our RSD value is only 6 on an 18-bit scale, which is really, really low, okay? And next, we also wanted to measure the linearity of the instrument. And so we measure the linearity by calculating the ratio of hard dye beads of different intensity at the different MFIs. So x-axis here being the MFI values of the beads at different intensity. You can see that our percentage variation, even though of our high MFI value ones, are only at about plus minus 2% across all of the detectors in our 3 laser system. And another feature of our instrument is that our resolution is not impacted by flow rate. So when you're running flow cytometers, they have often told you that you have to run at as low a speed as possible and not to run at a high speed. Mainly because a lot of other systems, they use a Gaussian profile for their laser beam, meaning that your peak or your stream, your core stream where you have your samples, would have to hit exactly in the tip of the laser beam in order to get the maximum signal. And so when you run on a high speed, what happens with the core stream or your sample stream is that it starts to vibrate, and that vibration would mean that you would hit the side of the laser beam where it's not optimal powered, and gives you a high percentage RCV and giving you an increased CV in your different signals. So what we have done to overcome this is by having a flat top beam profile. So meaning that even if we are running at a high flow rate, we can see that even if the core stream is vibrating, it will still hit at the maximal excitation and give you the maximal signal for your sample. So you can see that when performing at low, medium, or high flow rate, we do not see a loss in resolution of our 8-peak beads across multiple different channels. So this is very important for users who have a lot of sample and would want to run at a high flow rate, but also being able to avoid losing resolution. So next we're going to talk about some of the advantages of spectral cytometry and why people are moving towards spectral cytometry or full spectrum flow cytometry. Full spectrum flow cytometry enables us to use highly overlapping dyes. So like I told you, my favorite example being APC and AF647. You can see here in a conventional system, if you are only using one filter to identify the fluorochrome, you would mean that you are not able to separate out these two dyes because they are too close in terms of emission. However, when you're looking at the aurora, where we are looking at a full spectrum signature of the dye, you can see over here that in the regions that are circled, the APC and the AF647 have completely different signatures in terms of how the patterns look, in terms of how the peaks are looking as well, are completely different. And all of these differences allow the software to pull out CD8 APC and CD57 Alexa Flora 647 in the same panel, in the same sample. So you can see that we're able to pull out co-express markers in the double positive, even though we're using a very highly overlapping dye. All right. Another feature of full spectrum is that you are able to actually subtract autofluorescence from your cells. So in conventional systems, you're often told that the autofluorescence will need to be adjusted by pushing the negative peak down to 0. But over here, what we do with full spectrum is we identify the full spectrum signature of the unstained, which is also has your autofluorescence, as a signature, and we can extract out this signature from your sample. So in this example, you can see of E cells where you have unstained looking very high autofluorescence signature, very high MFI signals over here. And then compared to a CFP single stain control, it has very similar background or very similar signal, which is why without autofluorescence extracted, your negative population actually gets misidentified or wrongly identified as a CFP positive cell, when in fact it should be at 0 point here after the autofluorescence is extracted. So being able to extract your autofluorescence would improve your resolution of your data and also make your data plots more suitable for publication. Yeah, so it's very important that you improve the resolution of your data and also have data that is publishable and not looking like this, where it is, misidentified or wrongly identified as a positive population. So we talked about some of the advantages. I'm going to share with you how we have helped our users simplify their workflow for flow cytometry. So one of the things that we have done is looking at reusing the controls. So what we have done is we have taken a day 0 sample and prepared a day 0 sample, multicolor sample with single stain controls that we have prepared at day 0 as well. And then for the subsequent days, we actually did not prepare any single stain controls. The user just prepared their samples and they ran it on the machine without having to run any single stain. All right, so you can see at day 0, this is how the data looks like. So this is our standard or our mark that we are using, benchmark that we are using for how our data should look like. So you can see that the CD4s and CD8 are clearly resolved in our sample at day 0. We can isolate NKT cells, we can isolate Treg cells, we can pull out CD4 subsets and CD8 subsets and B cells as well from our single stain controls that we have prepared fresh and our sample that we have pulled out all of these populations. At day 2, we didn't prepare any single stain controls. We only prepared the sample, the multicolor sample, and you can see that our data is very comparable, even though it's been 2 days since we ran the instrument, and you can see that there is CD4 and CD8 still well resolved. CD4s and CD8s here as well, the subsets are well resolved. We are able to isolate Treg cells. And then fast forward to 77 days later, you can see that we are still able to pull out CD4s and CD8 able to isolate the Treg cells, able to look at the subsets of CD4s and CD8 T cells, as well as isolate B cells. So allowing our users to reuse the controls and showing them how stable the data would still look, and saving them the time and effort and materials to prepare the single stain control, especially when you have very, very little samples or very, very little cells. You might not have enough cells to run your controls. This is a great way to reuse your controls from a previous experiment or previous day. We also show that, you know, we are able to reproduce the data that has been used on multiple different instruments to get you the same data set. So it is good for multi-center trial, people who are looking at doing a large cohort study in multiple locations. You can see that across 4 different instruments, the same data being run gives you a MFI CV percentage of 5% or less across multiple different instruments. So this is important for people who are planning to run the same sample or the same kind of sample at multiple different sites or on multiple different instruments. So we talked about you know, what full spectrum flow cytometry is. The next thing I want to share with you is how our system compares with other conventional cytometry systems. So in order to compare the same thing, because we can run 40 colors, but a conventional system probably cannot match up to what a full spectrum analyzer can do. So we have to trim it down to a 21 color panel where there are no highly overlapping dyes and there are no you know, complex fluorochromes that cannot be used in combination. And you can see over here that we are able to use 21 colors with multiple different markers across, looking at B cells, T cells, NK cells, to look at the different subsets in a 21 color panel. So how do we compare the data that we generate? By looking at something we call the spillover and spreading matrix. So usually how we compare 2 different instruments is by looking at the amount of spread that is contributed to each instrument. So as you can see over here, the top row being the Cytek cytometer and the bottom row being a conventional cytometer. The more red the cell, it means there's more spread. The higher the numbers here, the more red the cell will be. So you can see right off immediately is that the Cytek cytometer has less spread relatively when you are looking at a compared to a conventional cytometer. So there's less spread when you look at the numbers itself. How about when you look at the data, when you look at the flow cytometry data, do you also see less spread? Yes, you can see here the top row being the Cytek cytometer and the bottom row being a conventional cytometer, that there is more spreading as compared to the Cytek instrument. The more spreading that you see over here across multiple different plots, leading you to resolution loss in conventional systems because there is more spread. And as you can see over here, both data have been scaled identically to make sure that you are seeing the same thing in both systems, so that you can see that you know, the scaling is the same, everything is fair and equal, and you still see a lot of spread in the conventional systems, and the spread would lose out on your resolution of your system, being able to pull out the positive and the negative populations. All right, so there's less spread in the Cytek instruments, leading to higher quality data, better separation, ease of gating your populations, easier to do your data analysis as well. All right, so now we come to the more exciting part about how we have actually used the full spectrum analyzer as for COVID-19 applications. So when the pandemic first started in February or in January, a a lot of our users approached us and wanted to know how can they use full spectrum flow cytometry to run their research projects or to run their vaccine trial on how to work with COVID samples. So we actually work with a lot of different sites worldwide in collaborating with them to evaluate if they would share the data and work with us. So we wanted to know that these places that were working on COVID would share the data with us and work with us, and we can provide a lot of different resources for them. We provided SOPs, things like standard operating procedures. How do you process your cells? How to titrate your antibody, how to run your staining protocol, some of the panel design as well. We have a lot of people, our team has a lot of people that are really great at panel design, being able to help them to design a 35 color panel, 40 color panel, to help with their research questions that they want to answer. And then as well as providing data troubleshooting and analysis expertise as well, how to look at the analysis, because you are running 35 colors or 40 colors, it is very different to run manual gating or just to gate on a population and look, and it is best to run automated gating or automated clustering to help ease the gating or data analysis workflow, and you don't introduce any human biasness into the data analysis as well. So we provided all of these expertise and all this information to our users who are interested in running a COVID-19 sample on the instrument. And so we designed a 35 color COVID-19 panel where we had a whole range of markers looking across B cells, T cells, DCs, dendritic cells, monocytes, looking at NK cells, Treg cells across a lot of different subsets of cells. We use very highly overlapping panel of fluorochromes like APC and AS647, per-CP, Psi5.5, per-CP fluoro710, BV421, 436, and E-fluoro450, and as well as things like 711, 750, and BB785. So all of these very highly overlapping dyes all put into one channel, into one panel, to give us 35 different markers to analyze on a cell. So this is some of the data or part of the data that we have pulled out by looking at this COVID panel. So we can see that over here we are able to look at CD4s versus CD8. So even though this is a highly overlapping panel where we had a lot of dyes which you cannot use in a conventional system, you can easily pull out the CD4s and the CD8s over here, and we can also isolate Treg cells using CD25 and CD127. And not only that, but we can actually look at the subsets of Treg cells. Being able to isolate the different subsets of Treg cells is very important as well when it comes to deciphering the immune response. We were able to look at CD4s and CD8s over here, isolating the naive, the central memory, and the effector memory CD4s and CD8 subsets within the blood sample. And then as you can see, all of our populations were easily gated. The positive and negative population are easily gated. And you can see that our negative populations are tightly clustered around 0. Giving you very, very nice FACS plot. We can isolate the B cells, the monocytes, as well as the TCR gamma delta T cells as well. So all of this information gave our researchers a lot of data to work with. They can isolate so many different subsets, so many different cell types for them to study. So what do we actually have to do when we're looking at such a huge data set where we have so many different markers, and how do we know what to look for and where to look for? This is where we introduce unsupervised clustering or very automated data analysis, and we perform this in the OMIP software, We check for scaling issues and then remove doublets, debris, dead cells, and as well as remove CD45 negative events. And once we have a clean population, we use something called UMAP, which is a dimension reduction software or algorithm, and then FlowSum, which is a clustering tool to then look at how the resulting clusters would look like. And so we can see over here, when using automated analysis, that this is a dimension reduction or a reduced way of looking at such a huge data set. You can see that we are able to pull out a subset here, a subset here, 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 and here. All of these clusters represent individual subsets of cells. So for example, in this cluster here, we actually are the monocytes cluster. So we have the classical, the intermediate monocytes, and the non-classical monocytes. We also have be able to pull out the CD4 T cells here, where we have our naive, our central memory, effector memory, as well as our Treg cells over here as well. And then we have over here our CD8 cells, our CD8 effector memory, naive central memory cells. And over here, these are our NK cells. And finally, over here, these are our B cells here and their different subsets. So just right off immediately after looking at this plot, you can see all of our different subsets have clustered separately and differently. And when you overlay this plot with like a patient or a severe COVID-19 patient sample, you will immediately see the difference between a healthy donor and a severe COVID-19 patient. You can also overlay that of a patient that has been given a vaccine and a patient that has not been given a vaccine to easily determine where you see differences in marker expression or subset expression. There's also the heat map of the FlowSum clusters, being able to show you where are the different subsets that you have, the CD8 effector memory cells, what kind of expression across all of the different markers in your panel, so that you can easily see that this is our B cell marker, is our B cell cluster, and these are our T cell clusters because they have CD3 expressed. So all of these gives us very clear visual way of analyzing the data and reading the data as well. All right. So since we have worked with our users since February or January this year, we have seen a whole lot of publications come out where they have used our SciTech instruments to publish this COVID-19 data. So you can see over here that on the left-hand side, these are the papers that have been produced in human studies. And on the right-hand side here, these are the studies that have been done in mice. So it has been used in both human and mice study, looking at not only just immunization or vaccine work, but also looking at evaluating whether mice model is suitable for COVID-19 study, as well as some of the more interesting data where we actually can tell whether a patient would be suffering from a severe or a mild form of COVID-19 by just looking at their subset of cells. We also have some very interesting data on how after you have been infected with COVID-19, how you actually have some memory cells that have been that would persist in your body after a mild infection. And these COVID-specific memory cells are very, very functional cells to protect you or give you immunity against COVID-19. And how some critically ill COVID patients display lupus-like B cell activation when you're looking at extrafollicular B cells. So having such a large panel allows you to dig really deep into the patient sample, into the PBMC sample, to get as much information as you can from your sample, because blood is very precious. You don't have a lot of these samples coming through all the time. So in conclusion, I hope you are convinced that our Cytec products expand spectral cytometry multi-parametric capabilities. We have a superior performance when compared to a conventional cytometer. We have very intuitive workflows with easy to use standardization and setup tools, as well as being able to show you that we have outstanding resolution in a 35 color assay in our panel, and then as well as how we actually can help support our customers to not only help them answer their research questions, but to provide them with the tools that they need to push out great publications in cell journals or immunity journals, even during this pandemic time. So the last few slides, I just want to highlight, our Northern Lights, that we now currently have a promotion of our instrument, where we have 1 lasers, 2 lasers, and 3 laser system, where we have shown that our 1 laser can run 9 colors. Sorry, our 1 lasers can run 9 colors. Our 2 lasers can run 14 colors, and our 3 lasers can run 24 colors. So it's a great instrument, benchtop size instrument as well, good for a lot of different kinds of applications. And we also have our website, where we have tools and posters and video tutorials on our website. You can also download our 40 color OMIP paper. You can download our poster on 5 laser full spectrum, as well as YouTube videos and customer testimonials on how much they enjoy using our instruments, or how much easier our instruments are as compared to our competitors. So last but not least, I just want to let you know that we are working with Lab India to move full spectrum flow cytometry into India, to introduce full spectrum flow cytometry to India. And if you have any questions, please feel free to reach out to either me or Rahul from Lab India, and these are our emails there. If you have any questions regarding the presentation, regarding the technology, please feel free to reach out to us. Thank you so much.
Thank you, sir. Thank you for your nice presentation. It was not boring, especially for your slides, including or discussing about the instrument. It is quite unique, sir.
Yeah.
I have a question.
Yes.
In case of donor and recipient matching, how HLA test can help the probability to tell the probability with the help of flow cytometry? In few journals, we have seen that flow cytometry is used in HLA matching in between donors and recipients.
Yeah.
Yes,
So yes, you can actually do donor HLA matching studies using flow cytometry. There are kits out there that are manufactured by different companies that work on HLA typing, and you can actually run the samples to determine whether the recipient and the donor are matched HLA wise, to determine whether they are a good match for organ transplant, or they are a good match for any of the blood transfusions studies.
Thank you, sir. There is another question.
Yeah.
The audience, can any specific food item can change the result or can affect the result? Any specific food item or its component? Is there any probability, sir?
So, sorry, again, could you repeat that question?
Yes, sir. Any specific food item taken by the patient before the test, can it affect the result of flow cytometry?
If they are eating a specific food item, is that what you're asking?
Yes, sir. Any specific component which we may take before the test?
Yeah, I think diet plays a huge part in affecting flow results. Like especially if the patient ate before drawing blood and before they actually take their blood test, if they actually have very fatty food or they have very very heavy meal, what happens is that all of the oil and the fats or all of the would affect your immune cells in your blood. So they will affect some of the marker expression if you have a very heavy meal or a very fatty meal just before you draw your blood for any of these data analysis.
Another question, sir, from the audience. The time taken for result, when we are getting the result, time is varying from sample to sample. That is, blood only takes a few minutes. Semen will take a little more time, and bone marrow will take longer time. Which is the point, sir? Is the density of cell or anything else? Why the time is varying from sample to sample?
Yeah, so it all depends on the volume of cells that you have. For whole blood, you can just work with 100 microliter of blood and you get very nice results from your whole blood. You probably have a larger volume for both your semen or any other sample that you have. There is a limit, like flow cytometers usually can acquire 35,000 events per second max. And so if you have something that's very concentrated with a lot of cells, you want to dilute it a bit more. But like we have seen with our slides, even if you run at a high flow rate, you will not have a loss in resolution. So you can dilute your samples and run at a high flow rate so that you can get all the samples that you need for your data analysis.
Thank you, sir. Mona Lisa, have you any question in the chat box? Hello?
No, ma'am, there is no question. Chat box.
Then another question is, sir, is there any side effect after the test? Hello?
After the, sorry?
Is there any side effect, chance of side effect after the test? Or after the sample taken by the pathological persons, pathological labs? Is there any chance?
So I don't understand the question. Are you asking about after the? Are you asking about any side effects other than loss of taste in COVID, or are you asking about?
Actually, sir, blood sample taking is very easy, but it is more complex in case of bone marrow sample. Is there any chance of side effects, sir?
Side effects. So the way that they take the blood from bone marrow or from the vessel is different. But blood is blood. As long as the blood that is extracted out gets put into a stabilizing reagent, so something like EDTA or those kind of blood stabilizing reagents, you generally have a very good quality of the blood or the immune cells preserved when you have these stabilizing reagents.
Thank you, sir.
Yeah.
It's a nice presentation and a nice experience for us. It's very new to us also. Thank you, sir, for giving time and staying with us.
No worries. I just wanted to share with you some of the data for a 16 color panel on our instrument. So let me just share my screen again. So this is just how a typical 16 color panel looks like on the instrument or on the software. And we are able to draw our gates on the population of the cells that we want to look at. We can double click on the population to pull out the CD4, for example. and you can then right click on the axis to then look at the downstream of the software, like downstream of the cell of the subset, like CD45RA and CD27, to look at the downstream subsets. And we also have the ability to adjust compensation on the software. So being able to show how we are able to run compensation on the software like this, where you can click and drag the population to adjust for compensation. We are also able to pull out statistics of the sample and have an idea of a count per microliter, because we have a flow meter in the instrument. So we are able to give you an absolute cell count of your cell samples in the your cell numbers in your sample, and give you a count per microliter of each of your different subsets as you have gated them. And so you can then use it as like a without using counting beads, you can quantify the number of cells in your sample for any diagnostic use or any other applications that you may have. Yeah.
Thank you, sir.
Yeah.
Now the time for vote of thanks.
Yeah.
By Dr. Mona Lisa Banerjee. Yes, sir.
Thank you, Edmund Chua.
Thank you.
Okay. We actually go through your total presentations. Very nice presented by you. Thank you.
Yes.
Please, madam, you just ask to our sirs for vote of thanks. Okay.
Yes, sir. Dr. Mona Lisa will deliver the vote of thanks for today. Professor Rai is also with us now.
Thank you, ma'am, for giving me the opportunity. Good afternoon, everybody. We have reached to the end of today's session. First of all, I would like to talk about the great coordination among our organizing team, technical team, and advisory committee. I would like to thank each and every member. Next, I would like to thank our eminent speakers. Professor Somnath Roy, he is my direct teacher. Thank you, sir, for your engaging talk on vaccine. Your talk has given a great start to our webinar. Then I would like to thank Mr. Edmond from Cytec Biosciences, USA, for his awesome demonstration and talk on flow cytometry. Thank you, sir. Next, I would like to thank Dr. Manush Ranjan Bhunia, the chief patron of today's webinar, and the honorable president of the governing body of our college. Then I would like to mention the constant effort of our principal sir, Dr. Tapan Kumar Datta. He is the patron of today's webinar. Thank you, sir, for being with us as a backbone. Finally, I would like to give a special thanks to all the participants. Thank you all.
Thank you, sir. Thank you, Mona Lisa. Actually, the theme of this webinar is the brainchild of our principal, Professor Dr. Tapan Kumar Datta. Thank you, sir. We will see all of you again tomorrow. Thank you, sir. It's over. Today's session.